Well, hello, everybody. My name is Kevin O'Brien, and I'm joining you from the west of Ireland. And welcome back to another YR webinar. As you probably well know, we have these every Thursday at 7 p.m. UTC. That's Universal Coordinated Time, and we are live streaming on YouTube. Before I introduce our speaker, Koen is joining us from Belgium. We would like to sort of invite you to sort of say hello and tell us where you're from, because one of the great things about these live streams is to sort of see the extent of and range of the R community around the world. So if you're in the YouTube chat, just actually give us a hello from wherever you're from, like hello from Ireland or hello from Belgium and so on, because this is we always get a real sort of kick out of that. Just before I continue, actually, I'd just like to thank our sponsors, Jumping Rivers, who have provided ongoing support to ourselves and many other R user groups over the past couple of years. And we appreciate their support. So the jumpingrivers.co.uk. So let me begin. Let me introduce Koen van der Berge. And Koen is going to be speaking on, I'm going to read it here, interpretation of single-cell RNA sequencing trajectories using additive models for differential expression analysis. So, Koen, I'll let you take it away there. And I'm going to go on mute, and I'll leave you to it.
Thanks so much. Thanks for the introduction, Kevin. Let me share my slides. No. Okay. We should be all good here. So yeah, thanks a lot for the opportunity of talking about our work in the YR webinar series. I've been checking a few of your series, and I think it's a really great initiative, and so I'm very honored I'm able to contribute to the YR webinar series. So today I'll try and contribute to the webinar by talking a little bit about biology. And so I'll talk a little bit about kind of big biological data sets and the statistical tools, but also the software, the R packages we're using to interpret this type of data sets. So my name is Koen van den Berge. I'm a postdoc, and I'm splitting my time between Ghent University and the University of California, Berkeley. So these slides I'll be talking about are public, and I hope they'll be shared in the chat. And if you can't get your hands on the slides, feel free to drop me an email, and I'm happy to share them with you. So as an outline for the talk, I'll start by introducing you to what gene expression actually is and how we study gene expression through single-cell sequencing. And then I'll go a little bit more in depth into one of the motivating case studies for the tools that we'll be using. And those tools we'll be using, they'll actually use smoothing, which is a third point. We will kind of provide an introduction to smoothing within this talk, just to be clear about what I mean when I'm saying smoothing. As a fourth point, we'll talk about the methods we've been working on and we've been developing, and that allows people to kind of interpret these large-scale biological data sets. We're calling the method tradeSeq. It's a statistical method that's implemented as an R bioconductor package. And we'll show you tradeSeq in action using two different ways to interpret these data sets. And then as a kind of bonus point, if time allows, so I'll check the time when we get there. And if you still have plenty of time left, I'll talk about the fifth point, which is kind of a work in progress me and my collaborators are working on. So let's start with the introduction. So as humans, many of the cells in our body contain DNA. Actually, most of the cells in our body all contain basically the same DNA. And right now we know that the DNA contains about 20,000 genes. That's kind of the knowledge we've gathered right now. And a gene is nothing more than kind of a piece of the DNA that encodes for a specific piece of information. So here at the schematic here, we are seeing the DNA strand in gray, and the gene is flanked by these two kind of shaded region, right? So the DNA piece within the shaded region is something we call a gene. So it's nothing more but a piece of DNA. And a gene always encodes for a specific piece of information, and we can kind of transcribe this information. So here there's a protein that transcribes this information, that kind of decodes this information in a gene in a process we are calling transcription, because that's a biological term we're giving to the gene expression. Once we do that, we get, so this protein that decodes this information, it produces this messenger RNA molecule. And this messenger RNA molecule, it kind of functions on its own, but more typically it's actually translated into a protein, and the protein are the actual functional building blocks in our cells, right? So it's the working horses in our cell that, you know, allow us to transport molecules, to digest our foods and all that stuff. So when we are saying we are studying gene expression like the data sets we'll be working with, what we're actually studying is the concentration of these messenger RNA molecules. So if you observe a lot of messenger RNA molecules from this gene, then we would say that this gene is highly expressed. So why is it important to study gene expression? Well, as I've said, basically all cells in our body contain the same DNA, but still our brain cells are something very different than cells in the liver than our muscle cells, right? And a major causative agent of what makes these cells look differently, behave differently, and function differently is actually their gene expression, right? So their gene expression drives them to a kind of developmental phase during embryogenesis and now allows them to retain their function as adults. So how do we study gene expression? So let's say we start with a tissue here on the left. So every kind of blob here is a single cell, and the cells are colored according to the cell type. So here we'd say we have 4 different cell types in this tissue. And traditionally, we have done what's kind of depicted here in the bottom row, which we are calling a bulk analysis. So what we're doing is that we are kind of breaking up all the cells in this tissue. So we gather this piece of this tissue, we break up all the cells, we gather those messenger RNA molecules, and then we kind of sequence them and observe an average gene expression profile from all cells in this tissue. And obviously, if we do that, then the cell-to-cell heterogeneity is actually masked, right? We only observe an average across all these 4 different cell types, and we cannot really uncover which different cell types contributed to that average. More recently, actually, new technology and new wet lab protocols are allowing us to study gene expression at the single cell level, which is really a biologically very relevant kind of unit. And if we do that, so first what we do is that we kind of dissociate the cells from each other. And then we look at the gene expression data on the level of the single cells. And obviously, if we're able to do that, we're able to kind of uncover the cell level heterogeneity in gene expression. And in this case, we are able to disentangle the 4 different cell types that kind of compose this tissue. So how does a gene expression data set look like? Well, actually, a gene expression data set, I will call this a single cell RNA sequencing data set, because RNA sequencing is kind of the technology we're using to measure gene expression. And I call it single cell RNA sequencing because it's at the level of single cells. But a gene expression data set is basically just a count matrix, right? So in this talk, I'll talk about the random variable YGI, which is the gene expression for gene G observed in cell I. And how this count matrix looks like, typically, at least in the way we are working with these gene expression data sets, we have the genes in the rows, and these are typically 10 to 20,000 different genes. And we have the cells in the columns, which are typically 1,000 up to even several millions of single cells. So these can be really big data sets. The values in this count matrix are typically very low, right? And the way you should interpret these, at least with the more recent, what we are calling UMI-based data sets. So here, for example, we are observing a 5 for gene 2 in cell 2. And that means that for that gene 2, we have observed 5 molecules produced by gene 2 in cell 2. So the expression counts are typically quite low. And these matrices are typically very sparse, right? So typically over 90% of the values in this count matrix are equal to 0. That does not mean that that gene was not expressed in that cell. All it really means is that we did not observe any molecules for that gene in that cell, because the protocols we are using are still suboptimal. And it could be that gene 1 had molecules expressed in cell 2, but we simply did not pick them up, right? So you could have a 0 because we didn't pick up any molecules of the gene in that cell, even though the gene was expressed. Or we could have a 0 simply because there were no molecules in that cell. So there's some inherent random variability to these counts. So I said that the number of cells we're observing is kind of in the range from 1,000 to even a handful of million. And this is a plot made by Valentin Svensson that kind of aggregates a lot of single cell studies. So on the x-axis, we have the publication date of a paper being published on single cell sequencing, or publishing a data set on single cell sequencing. And on the y-axis, it's a number of reported cells in that study. And what we're seeing is that the first paper basically looked at a handful of cells, but right now we are looking at data sets. At the moment when this figure was created, the record holder was this science paper that actually sequenced around 3 million single cells. So these are really, really big data sets. So I'll introduce you in kind of more detail to one of the data sets. And this is a data set I'm working on that was produced by the Nye lab at UC Berkeley. And the Nye lab is really interested in the olfactory epithelium. So the epithelium is kind of the outermost skin layer, and the olfactory system is the system that's responsible for the perception of smell. So the olfactory epithelium in mice is kind of the epithelium, the olfactory system within the nose of mice that allows them to pick up smell and send those signals to their brains. So here on the right-hand side, you are seeing a schematic of this tissue. What's very important in this tissue are the green cells here at the bottom here, which are termed HBCs, or horizontal basal cells. These are really important for this tissue because these cells are actually the stem cells of this tissue. So they allow us to regenerate the tissue in special circumstances. Another important cell type here at the back are the SUS, or sus-tentacular cells. And these are cells, for now, we think their function is to simply hold the epithelium intact, keep it straight. And then we have the cells here in purple, which are the mature olfactory receptor neurons, right? And these are actually the cells that have these receptors that pick up the smell signals and process them to the brain of mice. So we can already see here in the schematic that kind of the production or the development of these purple olfactory receptor neurons is kind of a complex process, right? So it's a multi-step process that starts at the stem cells, at the HBCs, then produces in dark blue GBCs, then immature neurons, and finally mature neurons. And this is a process the biologists are very interested in. So in this data set, what they've done is that they have actually injured the olfactory epithelium in order to trigger the stem cells to reactivate, to kind of be activated and to regenerate the tissue, right? So they injure the tissue in order to activate the stem cells to regenerate, to repair the tissue. And what we'll see happening is that the HBCs will divide more and more to produce more and more stem cells to help in regenerating the tissue. But they will also be developing into these sus-tentacular support cells and into these olfactory receptor neuron cells that are able to pick up smell. So here at the right-hand side is, so in this data set, we have about 20,000 single cells. And here at the right-hand side is a three-dimensional representation of these 20,000 single cells. So in this representation, every point is a single cell. And for every point, we observe the gene expression of about 15,000 genes, which we have now reduced to 3 dimensions. And something I should mention here in the YR webinar is that the R programming language and the R packages developed by bioconductor developers, but also other developers, are really essential for processing these data sets. For normalization of this data set, we have used R packages. For reducing the dimensionality, we have used R packages. For doing the feature selection, selecting genes to use in this dimensionality reduction, we have used R packages. So this is something really that, this plot is something really where actually many developers contributed, because we have used several packages in order to get at this very nice representation of this data set. So every cell, as I've mentioned, is every point is here a single cell in this 3D representation, and we are cells by their cell type. So again, in green, we have the stem cells, right, which again, were the source cells of this tissue. And we see that these stem cells actually produce 3 different clusters of cells.
Koen, I think something's happened with your audio.
I'm sorry, can you still hear me? You can't?
It's very faint.
Let me see if I can fix that. Is my audio better now?
Better, yeah. That's great. Cool.
Okay, great. Okay, so in this 3D representation, we were saying that every point is a single cell, and we've colored the points according to their cell type. So here in green are the stem cells of the tissue, so it were the horizontal basal cells. And we can see that these stem cells actually kind of branch out into 3 different directions. You've got the large blue cluster here, which are actually the new stem cells, so the regenerated stem cells. We've seen that these stem cells will produce more and more stem cells, so they'll divide. The gray cluster are the sustentacular cells or the support cells. And then we have kind of one very long developmental path starting at the stem cell stage up until the orange cells, which are the mature neurons. And this is a very long developmental path, which is something we've already seen in a schematic on the previous slide. So based on this 3D representation, we can fit something which we're calling a trajectory. So a trajectory are the bold black lines we have drawn here in this 3D space. And a trajectory is kind of a proxy, a representation of the development that these cells are undergoing upon injury. So we know that they produce these 3 different cell types in this 3D representation. And we kind of represent those developments using this trajectory, which is represented by the bold black lines in this 3D space. So just to get the terminology straight, we will refer to a trajectory as a collection of lineages. So here we are developing into 3 different cell groups. So here our trajectory consists of 3 lineages. And we have fitted this trajectory using, again, an R bioconductor package called Slingshot. So once we fit this trajectory, what we're doing here at the bottom left in the schematic is that we are defining a pseudotime for each single cell. And the way we do that is, so this is kind of a 2D representation at the bottom left of a trajectory. And what we're doing is that we are projecting each cell onto the lineage. And then we are defining the pseudotime of that cell by calculating the length of the lineage from its starting point up until the projection of the cell, right? So a pseudotime, the way you should think about it is if a pseudotime is low, then it means that that cell is very close to the stem cell stage. If the pseudotime is high, then it means that that cell is very far out in development. Okay, so let's start by talking about smoothing. So once we had this trajectory, the biologists were mainly interested in 2 different questions. The first one was which genes of these 15,000 we are observing are involved in development for each of those lineages. The second one then is whether we can recover the transcription factors responsible for the driving of gene expression. So a transcription factor is actually a protein that regulates this gene expression. So the interpretation is kind of a level up. It's already looking at the regulation of gene expression rather than the gene expression itself. But let's start with the first question. So here, what I'm plotting here is only the cells that were involved in the development from the stem cells, again in green, to the neuron cells, which are now in blue. On the x-axis is the pseudotime. So again, low pseudotime is close to stem cells, high pseudotime is close to the mature cell type, to the mature neurons. On the y-axis is the expression of the gene. So it's one of the genes in this data set. And what we're immediately seeing here is that actually the gene expression data is kind of very smoothly along pseudotime. And if you think back about the first question of the biologist, which was, how can we find the genes involved in development in this lineage? well, then it kind of makes sense to kind of estimate an average smooth gene expression function as a function of pseudotime and use that to infer, to check which genes are kind of changing in average expression along this developmental axis. So before we do that, I'll briefly explain the concept of smoothing. So let's say we are working, with the gene expression data of gene G in cell I, and we'd like to estimate a function, a smooth function of that gene that's a function of the pseudotime of Ti. So how can we parameterize this function, right? So how can we parameterize this f, which is just some smooth function? There's actually many ways possible. So one option is to basically use a polynomial function. So we can say fgt is actually a polynomial. It can be a cubic function or a quadratic function. The other way is to kind of define basis functions as a simple building block to estimate a smooth function. And these can be linear basis functions, or these can be higher order, for example, cubic basis functions. Very often additive models, which are models containing smoothers, are actually parameterized using a number of basis functions. And this parameterization is often referred to as a basis function expansion. So what is such a basis function expansion? Here, I'm again plotting the gene expression of this gene. And at the bottom here, I've plotted in these kind of dashed lines, I have plotted linear basis functions, right? I'm calling these basis functions linear because they kind of linearly increase and then linearly decrease, right? And since these look like tenths, I'm calling this the tenth function. So every color here is a different basis function that's increasing in some region, then decreasing, and then it's zero everywhere else. So here we can actually view each basis function or each tenth actually as a covariate. And we can simply estimate the contribution of that basis function using a simple linear model, right? So here, what we're doing here is that we are estimating a covariate, a parameter for each covariate, meaning for each basis function. And then we sum the basis functions multiplied by their regression coefficient estimated using a simple linear model to get the eventual function we'd like to estimate, which is our mean gene expression function. If you do this using linear basis functions, we are getting a linear piecewise function, right? And this is just because the function we are estimating will be nothing more than a weighted average of our basis functions. If we use more complex basis functions, so here in the left panel, we are using cubic basis functions. Well, now our basis functions are smooth. So the function we'll be estimating, which is the bold black line at the right here, will now be a smooth function, right? Again, because our basis functions are smooth. So what's important to remember here is that the function we're estimating is simply a weighted average of the basis functions. And if these basis functions are smooth, then the function we'll be estimating will be smooth as well. So then actually a very natural question starts arising, which is how many basis functions should I use? How many of these building blocks should I be using? And asking this question actually boils down to the bias variance trade-off, meaning that if we fit too many basis functions, which is here on the right-hand panel, then our function will be very variable, right? We can see it jumps up and down. We can see that it's actually too variable to be good. But if you use too few basis functions, and then we actually might miss a few patterns in the data because we simply can't capture them with this limited number of basis functions. So this bias variance trade-off is very prominent here. If we have too many basis functions, we will have a low bias but a high variance, and the reverse is true as well if we use too few basis functions. So in smoothing, we kind of try and overcome these issues. And what we're doing there is basically we're taking a reasonable number of basis functions so that we can overcome a large bias. But then we avoid having too much variability in the function. We avoid overfitting by penalizing the wiggliness of the curve, right? And very often we are actually penalizing the second derivative of the function, meaning that the slope can't change too much, meaning that the mean will be smooth of the function we are estimating. And the objective function, we would be minimizing then in order to fit this function. If X is now our matrix of basis functions, would be a least squares criterion that's penalized by the second derivative. So here S
is kind of what we're calling the smoother penalty matrix, and beta S beta would correspond to the second derivatives of this function. And you can see that we have this penalty parameter here, lambda, which kind of defines how much do we penalize smoothness. If we're saying lambda is a low value, we see that the function is still pretty variable. If we're saying lambda is equal to a very high value, then the function will be very smooth, right? So now lambda controls the smoothness of our function, and we can actually estimate this using efficient algorithms. So how do we do smoothing in R? So in R, I definitely recommend you to use the mGCV package, which is a very powerful, very flexible R package developed by Simon Wood for estimating smooth functions. And here are just a few quick examples of how you could estimate smooth functions in R. So we are using the GAM function to fit a generalized additive model. And then you have kind of the linear model formula. So we have the response data, that's a function of X. And we want this to be a smooth function, which gives us this S formulation. We want this to have 6 basis functions, 6 knots. And we would like these basis functions to be cubic regression splines. We can even have interactions. So here at the second line of code, the smooth functions of X are interacting depending on the value of C. So this is an interaction between a smooth function and another covariate. And we can fit kind of generalized additive models using any family of any distribution of the exponential family, for example, a Poisson distribution. So these are very, very efficient functions and very powerful for the analysis of these data sets. So if you'd like to know more about smoothing, these 2 books have been really instrumental to me. And if you'd like to know more about it, I think they would be very useful to you as well. So the left book, the semi-parametric regression book, also has a website with our code. And the generalized additive models book is a book written by Simon Wood, who developed this mGCV package, also contains a lot of our code. Okay. So as a 4th part of the talk, we'll kind of introduce the model we have been developing to kind of interpret these gene expression data sets, which uses smoothing. And then we'll show you how we use that to interpret these data sets. So we are calling our statistical model and our software package tradeSeq. And what we're doing is that we are estimating such a smooth function. Remember, the data set we were looking at, it was this trajectory with 3 different lineages, right? So with the development of stem cells to 3 different other cell types. And what we're doing is that we are fitting a smooth gene expression function from the development of stem cells to each of those cell types for every gene we are observing. The statistical model can be defined as follows. So remember, the gene expression data we were working with were actually counts. So here we are assuming a negative binomial count distribution on the counts gene expression data. We are modeling the mean of that negative binomial count distribution using a log link. And then the linear predictor consists of these multiple terms. And I'll walk you through them term by term. So the last term is something we're calling a normalization offset. It's just something that's very typical for sequencing data where we need to normalize to make the cells comparable. We allow you to model the mean as a function of covariates, like this could be cell level covariates, this could be patients we would like to correct for, maybe the cells were coming from different patients or other technical effects. And then the actual working horse that does the smoothing is this first term here. What we'll do is that first we'll assign cells to these lineages, and then we will estimate a smooth function of pseudotime for each lineage and each gene. The way we do that is using a basis function expansion. And this is why we first had this kind of introduction to smoothing, because this is a basis function expansion that allows us to estimate this smooth kind of function of gene expression as a function of pseudotime. And a very nice thing of these basis function expansions is that if you want to know which genes are involved in development for one of those lineages, then we can focus the statistical inference on the parameters of those basis functions. And I should say that we're building on top of the R mGCV package to fit these additive models. Okay, so let's take a look at some data again. So this is again the JUMP gene we are looking at. So again, the x-axis is pseudotime, the y-axis are the expression counts. But now we are looking at the data from all the different lineages. So we're no longer just looking at the neuronal cells, at the neuronal lineage, but we're looking at the data from all the lineages. So now we have, again, the development up to the neuronal cells, but also to the HBCs in gold and the sustentacular cells in orange. So if you fit these smooth gene expression functions, what we're getting are these bold black lines, which are our estimated smoothers, right? We can see it in the neuronal lineage, the expression decreases and then it goes up again. While in the other two lineages, when we produce new stem cells or sustentacular cells, the expression kind of goes up quite dramatically at the mature cell type stage. So, and this is simply using an R function fitGAM that's implemented in our tradeSeq package. So how can we use these smooth functions to kind of infer which genes are involved in development for each of those lineages? Well, we do that through something we're calling differential expression analysis. So that the name kind of suggests it. So we're looking for genes with different expression along development or between different lineages. So we have a range of tests we are implementing based on these smooth functions of gene expression. And the first set of tests is what we call within lineage comparisons, where we only look at differences in gene expression within a particular lineage. And so I'll mention a few of those tests, and the plot at the bottom of those tests, so I'm plotting here at the bottom of the slide, is kind of like the most significant gene we're getting at with that particular test. So a first test is what we're calling the start versus end test. And it really checks whether there's different average expression at the starting stage of each lineage versus the end stage. So here we can see that for all three of the lineages, the average expression is very different at the starting stage versus the end of the lineage. The association test here at the right-hand side is kind of a generalization of that. So we're no longer comparing two particular points, but we are really looking, is gene expression significantly varying as a function of pseudotime, or is it smoother flat, right? So that's how you could interpret the null hypothesis. And again, the most significant gene is something that is a gene whose gene expression actually varies, very drastically along pseudotime. A second range of tests is something we are calling between lineage comparisons. So the first test, the diff-end test, is something we're using to compare gene expression at the endpoints of each lineage. Again, the pattern test is kind of a generalization of that, looking for differences in the smoother, differences along development in average expression pattern between different lineages. Okay, so that's kind of what we have been using to answer the first question of these biologists, which was which genes are involved in development. The second question concerned these transcription factors. And so again, transcription factors are these proteins that regulate gene expression. And the way you could think about these is that somewhere along development, some transcription factors, definitely not all transcription factors, get activated, and then they will regulate gene expression of a few genes.
Hi, folks. I think we have frozen there. I'll just give a second there to see what we can do. We're back, are we?
Yeah, it looks like I disconnected for a while.
All right. Anyway, it's all part of the fun. I'll let you get set back up there, okay?
Yeah, sounds good. Okay. Sorry for that. So a second question of these biologists was looking at these transcription factors. So transcription factors are kind of genes regulating the expression of other genes. And you could think about these transcription factors as proteins regulating gene expression at a specific point in time. And once they've done their job, they kind of get deactivated again because the cells have moved on. And the way we're doing that is that we're again looking at these smooth functions of gene expression along pseudotime. And right now, if we kind of want to find out what we're calling a transcription factor activity cascade. So which transcription factors are active at which specific points in time. And we can then use that to kind of order transcription factors based on their activity pattern. So if we think about what we should be finding, that we should be finding transcription factors that kind of increase in gene expression, right? Because they're getting more and more active. And then we would kind of order transcription factors based on where they are most active, meaning where they are peaking in gene expression, and then they are declining again. And the way we're doing that is that we're using these smooth functions to look at their first derivatives. So their first derivative is basically the slope of the smoother at a particular point in time. And this is, for example, an example again for the Jun transcription, for the Jun gene, where we are estimating the first derivatives for each of those three lineages. So here, for example, if you look at the dark blue, the neuronal smoother. So here at the top right panel is the first derivative of that smoother. And we can see first it decreases a little bit. So the first derivative will be negative. Then we have a point at around pseudotime of 5, where the first derivative is basically around 0. And we see that actually the smoother indeed is kind of flat. So it's not really going up or down. Then after that, we have a very negative first derivative because the mean expression is kind of decreasing, and then it's increasing again, which is again showing in an increasing first derivative. So the first derivative is just a slope of the average function. And so if we go back to these transcription factors, what we're doing is that we are first checking for which transcription factors do we have a significantly increasing first derivative, right? Meaning that these are increasing in gene expression. And then we define a peak of expression for a transcription factor as kind of the location where the first derivative switches sign, which means that it stopped increasing and it starts decreasing, right? And if you do that, then we see that, so here I'm plotting a heat map where every row is a transcription factor. And the row, so the kind of x-axis here is the dimension of pseudotime. So again, on the left-hand side is expression at the early stem cell stage. On the right-hand side of the heat map is expression at the neuronal stem, at the mature neurons. And so every transcription factor is a row, and we've colored the rows in terms of the scaled gene expression of that transcription factor. And if so here we've selected only the transcription factors found to be active in the lineage, and we order them according to their expression peak. And we can see that we have this nice subsequent activation of transcription factors, this kind of nice transcription factor activity cascade that allows us to order these transcription factors and actually group them in terms of where they are active in the lineage. And this is very useful to understand the biological processes that are involved in development. So the transcription factors driving these differences in gene expression along development. Okay, do we still have time for a last part, or should we move over to the questions, Kevin?
No, go on for it. Yeah, go for it. Yeah.
Okay. Yeah. So as a last part, it's kind of a bonus part. It's kind of work that's still very much in progress. And the funny thing here is that, you know, our R package is also on GitHub. And someone posted an issue on our GitHub repository saying, oh, so how should I analyze a data set if I have multiple conditions? And this is something we're currently working on. And so I'll briefly mention a very small case study we're working on. I won't go into too much detail, but basically you can look at, we're looking at a development. Now it's kind of one lineage, so it's development of one cell type to another cell type. But we're observing this development in two different conditions. We've got a control condition and we've got a treatment condition. If you do a dimensionality reduction, right now it's in 2D space of these two conditions, we can see that these two conditions kind of basically completely separate, right? So if you like to know which genes are different in development in condition 1 versus condition 2, it's kind of hard because these conditions kind of completely not overlap, right? They're completely separate. And in order to kind of tackle this problem, we again need to think about kind of the data analysis, and we use different R packages and that implement complex algorithms that actually also allow us to integrate different data sets. So here is again a 2D representation of the same data set, but now we have kind of integrated the two data sets using an R package that's called Seurat. So it's a very popular R package for single cell analysis. So here on the left-hand panel is again, every point is a single cell, and cells are colored according to their treatments. And now we fit one lineage because we have only one kind of developmental axis in this data set. And then we are kind of using this one lineage to check how do cells develop differently between different conditions. So this is a simple density plot of the two conditions represented by the two colors. And you can see that in one condition, we have three modes, right? So at a low pseudotime, an intermediate, and a high pseudotime. While in the other condition, kind of the first mode of this density function is being skipped. So it seems like the cells develop faster once they are treated with this particular treatment as compared to a simple control condition. And then again, we can use these smoothers to kind of identify genes that are doing something different between these conditions along development. So before I finish, I think it's important here, I kind of acknowledge that all these plots, all these analysis really build on the R and bioconductor infrastructure, which is so bioconductor is kind of a software repository for R packages and other software, but mainly R packages, analyzing genomics and other biological data sets. And in particular, there's really a nice community attached to bioconductor. So they are writing a lot of vignettes to explain how to use the code. And they have actually also written books. For example, there's a book associated with this paper that allows you to kind of take a deep dive in using R for analyzing these single cell data sets. So it's a very nice community, something I definitely recommend you to check out. So right now, the latest bioconductor release consists of about 2,000 different R packages. So it's a really nice thing to check out. So thanks for your attention. I'm sorry if there was a kind of technical issue here, but I'd like to end this talk by thanking the tradeSeq team I've worked with to develop tradeSeq, so Hector, Kelly, Sandrine, and Lieven, the Nile Lab who produced these data sets and helped with interpretation, and also the Dinverse team at Ghent University. So if you'd like to know more about smoothing, I also provide the link here of Simon Wood's website, who's got a lot of documentation on smoothing, and 2 links to get started with the software of tradeSeq or read our paper. So if there's any questions or...
Hi there. Yes, we have a few questions. So you can leave the slide deck up there. Okay. So the first question is, I'll actually ask them in chronological order. Folks, if you're watching at home, you can put in a few questions there. We have a good bit of time left. So TP Flow Specialist is asking, what visualization tools does Bioconductor use?
It uses a lot of, so it builds on R. So bioconductor packages are typically R packages. And, it uses a lot of things. I mean, I guess, in R, you have the base R plotting functions, and you have the ggplot kind of community, like the tidyverse community. Yeah. So in Bioconductor, it's really just a mix. They don't push you to one kind of type of visualization or the other. And they also don't have like their own visualization. They're pushing forward. I think it's a very transparent community where really we're not pushing to kind of one type of visualization, but we're really pushing to the visualization that's most easily interpretable, and that's kind of the most kind of representative of the data that's underlying the visualization.
Good stuff. Just a second question is, and I'll go back to that actually shortly, but I have a second question here from Wanabi Falmoor. Now, I think you might have crossed over it after he asked the question, or they asked the question. Is k equal 40 an example of overfitting? So I think you might have covered it, but you could just talk about overfitting there again.
Totally. I mean, let me see if I can pull it up quickly, right? So totally, k. So here we are using 40 different of these linear basis functions to estimate this function, and we can see it's much too wiggly, right? This is not what we want, and so this is definitely an example of overfitting, because we're fitting the data very locally, because those basis functions are only active at a very tight domain of our x-axis, you could say. So here, this is definitely an example of overfitting. Yeah.
I might mute myself. Yes, good stuff. Thanks very much for that. A TP flow specialist again. Could you briefly explain how pseudotime is computed? Yeah.
So I maybe I haven't been totally clear, but so what we're doing to calculate pseudotime is so here we have this 3D space, right? And what we're doing is that we can project each cell to each of those lineages, right? And so we just take an orthogonal projection. And it's best described by the schematic here at the bottom left. And then we are looking at the trajectory, which is the bold black line in both of the plots. And then we're walking along this trajectory. So we're following the bold black line up until the projection of that cell. So let me annotate my mouse here. Okay. Oops. So, sorry. I'm screwing this up. Let me annotate. Okay. So here, say we're looking at this particular green cell, right? The way we define its pseudotime is that we project the cell to that lineage, right? Which is this point. And then we walk along the lineage until we go until that projection. And the length of this walk I've taken is the pseudotime of that cell.
Sorry, two questions now. I have two questions from Eugene Hickey. The first one is, why did you select the June B gene to study?
Oh, it's just an example gene. I mean, okay. We've got 15,000 genes in this data set. I found this a gene that was kind of nice to work with. It's actually a gene that's pretty important for the the data set we're looking at biologically. But many of the genes are could be used as alternatives. I mean, we've got a few genes in these plots here, and you can see every gene has got kind of a different gene expression pattern. But I just used the June gene as a kind of scenario to kind of make the framework and the data more kind of approachable.
Okay, yeah. And the second question from Eugene, do you have any thoughts on why the olfactory epithelium, or epithelium is it? Epithelium is one of the rare sites we see neuronal regeneration.
That's a really good question. That's a very good question. I think it might be a bit of a too biological question for me to provide a sensible answer to, I'm afraid. But it's a very interesting question, but I'm afraid I can't really answer that. I don't have sufficient biological knowledge of the system to really answer that question. Okay. It's a great question. Yeah.
Yeah, it's probably just out of scope. Maciek has a question. The expression versus pseudotime plots show discrete y-axis values. The horizontal lines, are the expression values binned, and why?
No, it's because we're working with counts, right? So counts are discrete.
Okay.
As I've mentioned, these expression counts we're working with are typically pretty low. So these are log +1. Like so here the expression is the count, and we do +1 to avoid taking the log of 0. So here on the y-axis is the log of the count +1. So this horizontal line at 0 is actually counts that were 0. Then we have the counts that were 1, 2, 3, 4. So it's really because of the discrete nature of the data.
Excellent. That's great. So I'll actually have a few questions. Just actually, first off, tell us about your own career path, actually, like undergraduate, so on.
My own career path is that, so actually I started off as a biologist, but really an ecologist. So I've always been kind of fond of birds, and I've done a lot of bird watching when I was younger. And so I actually started off as an ecologist, where I was actually, my research there was kind of focused on building a species protection plan for some kind of birds that were declining in Belgium. I'm originally from Belgium. And that was kind of the first real data set I've worked with. And ever since that moment when I was analyzing this data set and I cared about the output of these analyses, it kind of got me really excited about statistics and the power to use statistics to kind of increase biological knowledge, right? So it's really an essential tool if you want to do proper biological research. And so after I realized that, I kind of trained myself into statistics, and it drove me to do a PhD in statistics. So I've done a PhD in statistical data analysis at Ghent University, and which really concerned this type of data. So I was looking at gene expression data. It's still very applied work given my ecology and biology background. And during my PhD, I started collaborating with also with Sandrine Dedois at the University of California, Berkeley. And now I'm kind of doing a postdoc still at Ghent University with Lieven Clement, who was my PhD supervisor, but also Sandrine Dedois at Berkeley. So, and my research is still focused on kind of gene expression data, or at least large biological data sets. And the common theme here, I think, is building methods and building tools that we hope are useful to biologists, but also statisticians, to interpret these large data sets to increase biological knowledge. Yeah.
Good stuff. Just actually, as let's say, as advice to somebody starting out, what R packages would you consider they should look at? I noticed you mentioned mGCV, and that's one I'm very interested in myself. But let's see if you could give us a few more there, and just sort of like somebody starting out, what's the sort of good core skill set?
I mean, I think a very important part of being you know, whether you call it a data scientist or a data analyst or a statistician, I think a very important part is always data exploration. So always look at your data. Don't just fit a statistical model without looking at your data. So if you want to start out with R, the thing I would recommend is find a topic you're interested in. And grab a data set from that topic, and then kind of start exploring that data. Make simple plots, right? Making simple plots, maybe even clean the data, right? Data cleaning is also very essential to people in our field. Like, for example, we have cells that are of very low quality and they kind of distort the patterns we want to see. So we want to remove them. That's a way of data cleaning. But the most essential thing I would say is pick a data set you're interested in and just start exploring it, start making plots. And the way we are working is if we are working with a new type of data, with a new technology, we don't just immediately start thinking about statistical models, but we start visualizing the data first. That's always the first thing we do. Are there any particular patterns we see in the data? What's kind of the structure of this data set? Where are interesting correlations we might take into account? But I don't think I'm going to name specific R packages, because I think it's really context specific. I mean, here I have a lot of experience in genomics, and there I definitely recommend Bioconductor. But for example, someone who's more interested in looking at finance data, I have no idea which R packages that person should be looking at.
Oh yeah, no, I meant in the context of somebody starting out in your field, like Bioconductor.
Okay, so in my field, I think what I would suggest is look at the Orchestrating Single Cell Analysis Bioconductor book. So this is a really fantastic book. So it contains over 20 different chapters where they look at case studies, analyzing different data sets. So you can just follow the code. It contains both text and code. It contains very good argumentation of why we do each step, right? So why is it necessary to filter cells? Why is it necessary to reduce dimensionality to see structure? And that's definitely a starting point I recommend to anyone starting off in genomics.
Good stuff, yeah. I have a question here in the chat from Sergey. If you use the tradeSeq as a part of a pipeline, do you first find a trajectory using Slingshot, or other tools to find the trajectory, and then use tradeSeq to fit the model for different genes?
Right. So that's a really good question, and this is actually a slide I have skipped, but let me open it now. So this was a slide I was skipping, but. So there's many different ways to fit a trajectory. We are often working with Slingshot, but really it's just one of the packages that allow you to estimate a trajectory. And if you use, many people use tradeSeq just as one part of their pipeline to interpret their data set they have generated in the lab. So the tradeSeq fit GAM function, we actually allow input from a Slingshot object, but also you've got other packages like Monocle. Is a very popular package that allows you to fit trajectories. We allow you to have kind of a cell dataset object, which is a specific monocle object. We just input it in fit GAM and we are able to fit the models for you. And then we also have a kind of general structure where there's always new kids on the block, right? There's always new people developing more packages. So we also have a kind of general input where as soon as you have basically a matrix of pseudotimes for each cell, we can fit the models. But I think it's very important here to make your software you're developing accessible to as many people as possible, right? So we might have our preferences for a method to fit something upstream of what your package is doing, but I think it's really important to be open to what other people are doing.
Yeah. Oh, thanks for that. Good stuff. We've no more questions, but I have one myself, and it's a very broad question. This webinar is being recorded in February 2020, and we still have the COVID crisis going on. So the question really is, how are you getting on? How are you and your colleagues and peers getting on and coping in this crisis with your research, progressing research? Just a sort of general topic there.
I mean, I think it's. I think we are one of the lucky ones, right? Because we don't have to go into a lab to make our experiments, to generate data. We are the lucky ones who have our laptop and are able to analyze data on our laptop. And using our laptop, we can do basically anything our work requires us to do. So I've been coping pretty okay. I don't think I should be complaining. Obviously, the isolation, I think, is tough for anyone. And it makes us realize how much we need other people and how much we need to socialize with other people. So I'm happy I've been able to kind of contribute to these webinars, which I hope are kind of providing at least some sort of social interactions, and we can keep these things going. But I've been coping pretty okay, but I think also I've been with, I'm with the lucky ones. And I just hope this all blows over soon enough, just like anyone else. Yeah.
Good stuff. I think we'll leave it there. So I'd like to thank Koen for joining us. And also, just actually on the note he left us with, people, our users from around the world, if you're interested in doing a talk for YR, please get in contact with us. We would love to reach out. All parts of the world and all subject areas. We'd be very interested in getting, providing an opportunity to host you. So we'd leave it there. Thanks very much. Just actually once again thank our sponsors, Jumping Rivers, who provide ongoing support. And so next week we will be back, and there's again next Thursday at 7 p.m. UTC. We have our next webinar, which is R in production in the cloud. And so I think we'll leave it there. Thanks very much, Koen.
Great. Thank you.
And thanks everybody for joining us. See you next week. Bye-bye. Now, I think we